All right, why don't we get started? I had hoped to have a few more people, but that's okay. I'm going to record these sessions, and hopefully, if people can't make it, I got a few inquiries about people who couldn't make it, and hopefully they can make it to the recording. So, welcome to this inaugural meeting of DevoWorm ML. So, I know that you have questions about what we're going to do, so I'm going to do a short presentation. I'm going to share my screen. And again, this will be on YouTube and it'll be available to see. I don't know what resolution though. So, and I can share these slides with you. So here, let's go back. Here's the first slide. Welcome to DevoWorm ML. This is a new initiative we're trying out. Sponsored by the DevoWorm group. You can see the URL there. This is a group of people who works on issues related to development, and we're aligned with the Openness Foundation, so we're interested largely in C. elegans, but also broader questions of theory and of using other types of organisms to answer developmental questions. And then we also have DevoZoo, which is a series of open data sets that are public, that have been reanalyzed from other data sets. There's a lot of neat stuff there if you want to check it out. And that's, of course, relevant to what we want to do in this group, which is to, the ML stands for machine learning, and we want to analyze those data in some way, or make sense of data, maybe even make sense of theory using computational techniques. So why don't we start with introductions. I'm going to stop sharing for a minute, and I want you to give a couple pieces of information. If you can give your name, your level of education or affiliation, your area of expertise, whatever you think that is, and then name one thing you would want to learn from this group. So why don't we start? Who wants to start? Who wants to introduce themselves first?
Let the student start first.
Okay, how about Vinay?
Hello sir, can you hear me clearly?
Yes.
Yeah, thank you sir. So my name is Vinay Varma. So I am a final year bachelor student, computer science bachelor students from India. So I've also worked with over the last 3 months. I've been involved with GSOC and I contributed to the DevoWorm group. So, and I basically worked on computer vision based project. That's also my interest, area of interest. Like in computer vision, I want to pursue a career. So that's maybe I'll want to go for higher studies regarding that, pursue a master's regarding like involving machine learning and software. So that's that. And from this group, I would I always believe when we share our resources and when we put a collaborative effort, then I think it's good for everyone. Like everyone gets to learn everything, and I think this computation biology is also a great field, and which has very much potential for machine learning algorithms uh, to get nice to get sense out of data. So I think that's what I'm looking forward from this group.
Good. How about Jesse?
Hey, good morning or afternoon, everyone. It's morning for me. My name is Jesse Parent. I'm based in New York. I recently I went back to school and finished an informatics degree. So I'm interested in applying computation and technical techniques to studying a lot of things. I'm particularly interested in, let's say, complexity, or maybe an anachronistic term of cybernetics. But I'm happy to have been working with some of those things they came up in the orthogonal labs and the Bratenberg vehicle group over the summer. So I'm interested in continuing working with those things and exploring my own interest in complexity, and particularly within the field of cognition and things that contribute to cognition. So that's sort of an interest here, but I'm also interested in generally getting better at machine learning and understanding how to apply that to biological data. So I really like the lab and the approach. I'm particularly here because it's a unique place in terms of the interdisciplinary stuff that it lets you look at. So I really like that, and I'm looking forward to seeing where this project goes.
Thank you. Okay, now we come to Dick. Richard Gordon.
Well, I'm a retired professor of all sorts of things. My career on embryology, and my background's in math, chemistry, and physics. What else can I tell you? I have come to a conclusion that the embryo is indeed a cybernetic system. I didn't know that was an anachronistic term, but it's amusing. And that it's a very peculiar one, where one of the goals of the cybernetic system is to build more cybernetic systems. It's going to be very interesting to see how this plays out. The analysis you guys will be doing and have been doing of embryo development, see if we can come to a new view of how embryos build themselves. Because an embryo is a very strange thing. It starts generally as a little sphere with no symmetry, only spherical symmetry, and it ends up being a bizarre organism with all sorts of asymmetries, including us. Okay. Nematode is a nice example because we can talk about individual cells and what they're doing. So I very much look forward to this. In terms of machine learning, I am only a, how should I say, observer of what's been going on. I haven't done any myself, but I've done enough reading to be skeptical about accepting its results. Okay, thanks.
Okay, so yeah, we have a pretty broad set of intro. Oh, here we go. We have someone new here. So yeah, just in time. Hello, Abraham? Hello? So yeah, while he gets ready, I was saying that I think we have a pretty broad set of interests here. And I think it's interesting that we have a healthy skepticism of machine learning, or at least maybe, this is one of the things we can explore in this group is like, you see a lot of papers and they report these wonderful results, but, they're not, what do they really mean? And are they really as robust as we think they are? There's a lot of hype around machine learning, but really, what is it? Especially when you apply it to something like biology, what is the, what is the value of that? So, Abraham, are you there?
Yes, I am.
Hi, how are you? Well, we were just doing introductions, so I was wondering if you could introduce yourself.
Sure. Hi, I'm Abraham. I'm a grad student at Stony Brook. I work with Dave Mattis.
Okay. And so why did you come to the meeting? How did you find out about it, first of all?
I actually found out about it from my PI, who was reading. There was a posting on the Node webpage, which is how I found out about it. Yeah.
Okay.
Yeah. And I went, oh, that sounds cool. My interest here, I'm using some doing image segmentation in larvae using neural networks, actually.
Okay.
Yeah.
What organism?
Celiacs.
Oh, okay.
Yeah.
Okay.
Okay. Jesse's from, I think, Albany. So, yeah. So we have a nice group of people here, and we'll probably have more people coming in and going over the course of the group. So I'm recording this, to give people who aren't here a chance to interact. And so, welcome. I think we have a pretty diverse set of interests here. So I'm going to go back to the slides now. I'm going to present my screen.
Okay, I should have said I'm in the middle of Canada, north of Bradley.
Okay. Okay, so then we back to the slides. So we did the introductions, and so a little bit about machine learning. So there's, like I said, there's a lot of hype around machine learning, and so I just took this definition from Wikipedia. Machine learning is defined as the scientific study of algorithms and statistical models that computer systems use to perform a specific task without using explicit instructions, relying on patterns and inference instead. It is seen as a subset of artificial intelligence. So, there in that definition, people might start to have, might have a different definition. But I think that's kind of, in some sense, what we want to look at with respect to biology. Is, can we apply these types of methods to biology? And, is it useful? What are we getting out of it? So, and we have, some computer science type people, some biologists, some people who know something about the methods I'm going to talk about here, which is a graphical history of machine learning. So when we talk about machine learning, we're really talking about these things that you see on your screen. And it started in the 1800s with regression techniques. And, you've added especially since the 1950s, really the advent of powerful computers that have allowed us to incorporate a lot of these different techniques, like Bayesian methods. Even Turing's learning machine contributed to machine learning. And you have all of these things. The AI winter in the 70s really kind of drove a lot of artificial intelligence people to say, well, we need something that we can apply to problems instead of just building machines that can play chess. And so a lot of this sort of coalesced in the 2010s into machine learning. And so, my vision for this group is pretty broad. If you're interested in image processing, great. Maybe you'll be exposed to things like neural nets or, support vector machines or something more esoteric or deep learning. And those things, that's nice that we're able to learn about those things. But there are also things not on this list that probably have contributed to machine learning as well, and maybe those are things that we can discuss in the group. So a quick summary of machine learning. Basically, one of the sort of the models for machine learning is this idea of taking data, labeling it in some way, and then training things and then testing. So actually I have these labels mixed up here on this diagram. So you start with some data set, and it usually has to be fairly large, but it doesn't have to be. All right. Okay. You start with a data set here, and it can be fairly large, although it doesn't have to be. And you want to find patterns in the data, like you usually do with a statistical analysis. Then you can either use that unlabeled data, or what we call unsupervised techniques, or you can label the data in something we call supervised techniques. And so supervised techniques are maybe easier to do, but people have done a lot of things with unsupervised techniques. And so labeling the data means putting names on like maybe segments of an image or attaching functional information to some data point, things like that. And then when you get to your actual model that you're going to use for machine learning, there are two aspects to this analysis. First is the training period, which is where you take some data and you train the machine, or you train the kernel or whatever it is to look for patterns. So you give it like a fair amount of data, you give it as an input, and then the machine kind of learns the associations, and then it has a model to work with. And then at the end of it, you have this testing phase, which is where you're testing for consistent results with that training model. So this is the result that you get. And so this is the sort of thing when we talk about machine learning, this sort of, 3-level approach, where you have the data set or label data, you have this training period that you train the model, and then you have this testing period where you actually test the accuracy of your model on the data. And, there are a lot of caveats to that. So in biology, we have a lot of variation. And so sometimes, they don't work as well as we'd like because there are a lot of outliers. But more recently, people have done things like looked at adversarial approaches, which are to use different types of training data where you have maybe the norm of your data set and maybe some data that is, say, adversarial, have different properties. So, if you had hands, would you want to train the machine in only hands with 5 fingers? Or would you want to train it maybe on some images with 4 fingers or 6 fingers? There would be, adversarial examples in the training set. And then that would improve your accuracy on the testing set. So this is the kind of thing I'm talking about when I talk about machine learning as well. And I think a lot of you, if you read papers on it, you'll see what I mean. This is the basic approach, but this is only one type of approach we're interested in. Machine learning is kind of that name casts a wide net, and so it may or may not include a lot of the things I'm going to tell you about here. But we're also interested in things that go beyond maybe a traditional definition of machine learning. So there's something called reinforcement learning. And that's based on what they call behaviorism, which was something they used in psychology years ago, but has lived a very robust life in computer science as a stimulus response sort of machine learning technique. And so sometimes you'll run into papers like that. Well, we could talk about those. Generative models. Many outputs not directly related to input. So, if you think about something like a genetic algorithm or even a neural net, those are things where you put an input in and it generates a lot of potential outputs that you have to judge whether they're good or not. But the model that generates those is called a generative model. And we're interested in those things as well. And finally, and this is Jesse mentioned this earlier, we're interested in systems and complexity models. So things like dynamical systems, networks, and game theory, and maybe more traditional cybernetics. Those are things that are really of interest too. And, there are probably a lot of linkages between machine learning and those models, those types of approaches that are really interesting. So what kinds of applications do we have to developmental biology that we can think of? What kinds of, why is it useful to developmental biology? So one is image processing, which includes segmentation of images, classification of objects in those images, and pattern recognition and microscopy data. So you can think of a host of different applications in developmental biology, and maybe we can bring some of those to bear in this group, discuss them, see what we think about different papers, different applications, and so forth. Then, standard data analysis. So we have problems in developmental biology that require, some sort of analysis more than a t-test, or more than some sort of correlation thing, where you would calculate basic descriptive statistics. In this case, you might have a statistical model for looking at movement of things, or some regulatory process in the genome. So there are a lot of papers where people, in systems biology especially, where people have attempted to analyze very large amounts of, sequence data or gene expression data, and they're using, let's say, network models or other things. And machine learning is definitely one of those areas that, people have applied to these problems, and they haven't done it at such a rate that we really have good methods for them. So they're kind of just, people just publish, or at least they used to do this in the near past. They'd publish a paper, and it was just we applied this technique to the genome, some gene expression data, and here are the results. And we don't really know if it's a good result or not, or a good model. they're the only one who's ever done it. So one of the reasons why, maybe there's some replication problems with some of these studies is that, it really is like a first attempt. And so maybe we can consider some of those things as well. Finally, there are things in the area of modeling and simulation. So people have done, things that largely are independent of data. Algorithmic approaches to growth and form come to mind. So, you'll have like a model of like morphogenesis. Actually, I'm working with someone right now. He didn't make it today, but I know he's interested in the group who's interested in modeling morphogenesis. And so, one of those things you can do to understand morphogenesis better is to create a model that just has a series of rules attached to it. And then the system evolves by those rules. And then you can analyze those outcomes. And there's no data that's put into it. It's just a series of rules and an agent-based model that responds to those rules. And so, those are, maybe he'll talk about one of those types of models in the group. But that's something we're interested in as well. So I think we should talk a little bit about points of collaboration and openness. Now, I know this is maybe some of you aren't familiar with this stuff, but I wanted to give people a very broad set of interaction points for this group because we're all in different parts of the world or different places in space and probably in time because we don't have the same schedule. So one of the things that I like to do is I like to have, a number of channels where people can ask questions, contribute in some way. So one of those ways is through GitHub. We have a GitHub repository. I can send out the links to those, those resources where that repository is. And this is for those of you who are aware of what GitHub is, it's it's a place where you can submit things for groups. It's based on computer code, but we can exchange. We can do editing of documents in it as well. So it's a version control repository. So you have things that are in committed, what they call committed to GitHub, and those things then can be edited by anyone in the group, and you can contribute new things. We can also use Google Docs for that as well if we're working on a document. But that's one point of collaboration. Another is we have a Slack channel, and I think it was the Slack channel was mentioned in the email. If you're not part of the OpenWorm Slack, I can send you a Slack pass, which basically lets you into the team. And then we have this channel where we would have conversations. We can post links to things or papers, and we can talk about things sort of, in a group without really being present. Then, of course, we'll have YouTube for like meetings like this, or maybe tutorials or hackathons where we get together and we solve some problem. So then, if we can post that to YouTube, other people can see it who weren't able to make the event. And then finally, I'm using the Node. Abraham mentioned the Node. It's a blog for notifications, or essays. It's basically a publishing platform, and you can post things, and it goes out to the developmental biology community. So that's a good way to engage people in developmental biology on these methods. And of course, if you're interested in sort of, getting people involved in these methods, one way is to communicate with them, people who might understand it a little bit, but don't really understand it a lot. And, by engaging people in sort of the place where they go to look for things, you can get people interested. So I think that's, there are probably more places we can work, but that's my philosophy about working together. So we can work together in multiple ways. And so the structure of this group over time, and we can, move the meeting time if this isn't optimal for people. I had a couple of people say Wednesday didn't work, but we're maybe still working on that. But anyways, the structure of this group will be, we'll have a meeting, and then people might want to lead a discussion of some type. So people would choose a date to lead a discussion. So, you might pick a paper to review, or you might want to show a demo of something you're working on. It could be anything. It could even be just a document or a PowerPoint presentation if you think that our feedback would be useful to you. Or we can brainstorm ideas. If you're not really sure what you want to do or how you want to contribute, things sound interesting to you, but you don't really know how to proceed, then we can brainstorm in our group and give feedback. Also, we could do this weekly, but we might end up doing it bi-weekly. We might do like 1 week where we check in and report on what our progress on things, or if we have something interesting that we want to share with the group. So the meeting frequency is could be weekly for a while and then bi-weekly. It's up to people in the group. And then we want to, at the meetings, we also want to plan for outputs. So we'd ideally like to do something with this group. I mean, we'd like to talk and interact, but we'd also like to do things like maybe, review or maybe work on papers or blog posts or presentations, and then maybe even educational content where we can educate people, say in developmental biology, about different types of, machine learning techniques. Or conversely, we could, try to teach computer scientists a little bit of developmental biology in ways that they might not be able to encounter in a classroom. I've had a lot of people in our DevoWorm group, a lot of computer science people, ask about like things in development, and sometimes they're very good resources, and sometimes they're no good resources for these things. So to make like the biologically oriented people aware that like there's an opportunity to educate computer science people on something in developmental biology that really doesn't necessarily exist in digital form or some easily accessible form. So this is my contact email if you have any questions or comments. And then, do you have any questions for me? I guess is the next question. Anything you want to add or have me clarify?
Yeah, Bradley, what you're suggesting on the talks might be in some sense a reincarnation of the embryo physics course that we gave some years ago.
Yeah.
Okay, and you might add that we have those online. You might add those as one of the resources.
Okay. Yeah.
And by the way, we need to migrate them. If we want to keep them, we need to migrate them someplace. So maybe you could think of where that might go.
Where are they now?
They're with a fellow in Germany, his name escaped me offhand, who was basically our webmaster. And he'd like to stop being responsible for it and have us pick it up.
Oh, yeah, we can look into that. Yeah, I think that some of that might be interesting to people. Oh, thank you, Jesse. So Jesse said, no questions, but I'm excited about all of this. So yeah, I mean, again, like Jesse, I think we did this over the summer where, check around the node. That's an interesting place. They have a lot of good resources there. So yeah, I mean, if you have just a brainstorming idea that you'd like to explore, maybe like do a short presentation on the idea, like what's coming, what's in my head, and maybe pose some questions, do it in a little bit structured way, but definitely something where people can, like we'll have a machine learning person who might say, oh, that's a good idea or not, and be a biology person who says, well, that's, there's something in developmental biology that I know about that is relevant here. So, that's the thing about these interdisciplinary groups. You can get like a lot of different perspectives. So, yeah. So any other questions? about Vinay? You've been kind of quiet about.
No, sir, nothing from my side.
Okay.
I think everything is clear. Like, yeah, good.
Okay, thanks. How about Abraham?
No questions on my end. Things sound great. Yeah, I like these ideas.
Okay,
Yeah.
good.
Mm-hmm.
So I think I think we have a couple other people who are interested in the group, so I'll contact them, see what they're, and this might not be a good time for them, so we might end up, maybe moving the time every now and again to different days, or we'll see what happens with that. But why don't we plan on how about next week at the same time? Is that good for everyone here?
Mhm.
Yeah,
Okay.
works for me.
Okay. Um, okay.
Yes, sir,
Well,
good, sir.
well, like I said, we might move the time around over time. So, and then Vinay shared a link here. So this is the link here. He put it in the chat to the GitHub repository. So there's the github.com in the chat. DWML. So that's the repository I was telling you about on GitHub. And right now there's not much in it. It's just a little bit of information about the group. And then, but the idea here is we can post, code examples for machine learning, or we can post we can do brainstorming documents, and we can get into those at another time where we just like put together some literature review and some questions, and we put it in the same document. So it would be like a working document where you have a lot of resources in the same place, and then eventually we could hammer that out into something like a paper or some sort of tutorial. So there are ways to use GitHub for that. The other thing that Vinay posted is this second link, and that's an example actually of someone working on deep learning for, I think it's microscopy data, or it's a C. elegans related thing. So this is an example of deep learning in, I think it's a network science model. So I mean, this is like just an example of what people are doing in the area, and it's pretty, like, I don't know if you can really, if you look at it, you may or may not be able to understand what's going on in the repository, but this is something that maybe if we had a paper to review, the basic concepts, it would be, more useful to people when they go to a link like this, because you have a lot of people posting, data and methods on GitHub, but it's hard to understand what they actually mean. Uh, we'd like to make it so that people are more comfortable with these things. So, I mean, that's basically one of our goals. So, if you have like references, we can post them in the GitHub repository as well. So okay. Well, thanks for attending. I think we're gonna conclude things for this week, and I just wanted to have an organizational meeting, and hopefully we have more people joining our group and contributing, and hope that everyone has a good week. If you want to maybe talk about something next week or present a paper, let me know. You had my email address there. Let me put it in the chat. Here's my email address. So if you don't have any more questions, okay, yeah, thanks for attending. Okay.
Thanks for having us.
Yeah, thanks. Okay, talk to you guys later.
Bye.
Okay, bye.